Mitochondria. Yes, you heard that correctly. You might ask yourself, you mean the thing we memorized in middle school as the powerhouse of the cell? That system shouldn't exist? How? Let me explain. Right now, inside your body, trillions of tiny engines are running a marathon without rest. But here's the kicker. They aren't technically human. They carry their own separate DNA. They replicate whenever they feel like it, and they act more like a permanent house guest than a part of the family. We've spent decades calling it an organelle, but in reality, you are being kept alive by an ancient, microscopic foreigner that never left. It's the ultimate biological paradox, a system that is entirely independent, yet without it, you'd stop existing in seconds. Mitochondria are tiny structures inside your cells that take the food you eat and turn it into the energy that keeps you alive. At first glance, that sounds simple enough, almost boring. But the deeper you look, the more unsettling it becomes. This system does not behave like other parts of the cell. It carries its own set of instructions, separate from the rest of your body. It makes some of its own parts. It multiplies on its own schedule. And most strangely, it is passed down only from your mother, ignoring half of your family history entirely. It feels less like a tool built by the cell, and more like a guest that never left. What makes this feel wrong is that cells are supposed to be unified. Everything inside them is expected to follow one central set of instructions, one plan, one authority. Yet mitochondria do not fully follow that rule. They have their own genetic blueprint. They resemble bacteria in shape, behavior, and even in how they divide. It is as if every cell in your body contains tiny life forms that were never completely absorbed. Nature, as we usually imagine it, does not like loose ends. This feels like one. According to the basic rules of biology, this system should not exist. One living thing should not survive inside another while keeping part of its independence. That usually ends in disease or destruction. Cells are supposed to reject intruders or be destroyed by them. Cooperation at this level should be impossible. Two separate life forms, sharing one space without one taking over, should collapse into chaos. And yet, here it works flawlessly, billions of times over, in every human body. When scientists first began to understand mitochondria, the idea was deeply uncomfortable. The evidence pointed to something shocking. These structures were not built from scratch by complex life. They were once free living bacteria that entered another cell long ago. Instead of being digested, they stayed. Instead of becoming enemies, they formed a partnership. Many researchers resisted this idea at first. It sounded too strange, too messy, too much like a violation of how life was supposed to work. But the clues kept piling up, and eventually the impossible explanation became the only one that made sense. The implications are quietly profound. If mitochondria exist because of an ancient accident that turned into cooperation, then life is not just built on competition. It is built on fragile agreements that somehow held. Your energy, your movement, your thoughts, all depend on a relationship that should have failed. Every breath you take is powered by something that was never fully yours to begin with. It leaves you with a gentle but unsettling thought. Maybe life is not as clean or as controlled as we imagine. Maybe survival does not always come from dominance or perfection, but from unlikely partnerships that barely make sense. And maybe deep inside you, the story of who you are began with something that should not have worked at all. It is strange when you think about it, isn't it? We memorize mitochondria as if they're just another part of the cell, a harmless fact from middle school. But once you understand what they really are, ancient outsiders that became indispensable, you realize how thin the line is between what we call self and what we quietly rely on without ever questioning. You might have heard pieces of this mystery before, but stay with me, because this is only the beginning. Mitochondria are just the first crack in the story. They open the door to a long list of systems that behave in ways biology never intended, systems that work only because nature occasionally bends its own rules. And if this stirred something in you, some small feeling of discomfort or curiosity, stay a little longer, because the deeper we go, the stranger it gets. And some of the most unsettling questions are still ahead. Zombie proteins, prions. Most of us move through life assuming that the rules keeping us alive are stable and dependable. We trust that the same quiet systems that let our hearts beat and our memories form will continue working tomorrow just as they did today. Biology feels like a set of laws that cannot be broken. But every once in a while, nature reveals something that feels deeply wrong, something that quietly asks whether life is as orderly as we think it is. Zombie proteins are one of those things because they challenge the idea that life needs to be alive in order to cause harm. In the simplest terms, zombie proteins, also called prions, are proteins that spread damage by forcing other proteins to change shape. What makes this system so unsettling is that it behaves like an infection without being alive. There is no cell, no metabolism, no intention to survive. It does not breathe, eat, or reproduce in any normal sense. And yet it spreads. A single misfolded protein enters the body and begins touching normal proteins, quietly convincing them to twist into the same broken shape. One by one, healthy proteins are converted, not by force, but by contact. It feels less like biology, and more like a bad idea that spreads simply because it exists. At first glance, this should not be possible. We are taught that biological threats must be alive. Bacteria are living cells. Viruses carry genetic instructions. Parasites grow and feed. Zombie proteins do none of this. They carry no instructions, no code, no blueprint. They are just protein, the same basic material already present in your body, and yet they behave as if they have a goal. They move through tissue, spread from one organism to another, and slowly destroy the brain. Nature, according to everything we thought we knew, should not allow this kind of thing to exist. Under normal biological rules, information flows through genetic material. Instructions are written, copied, and passed on. Proteins are supposed to be the workers, not the messengers. They fold into a shape, do their job, and eventually break down. Zombie proteins flip this logic upside down. Their shape itself becomes the message. There is no instruction manual, only a physical form that teaches other proteins how to become broken. It is like a key that reshapes every lock it touches until the entire system fails. When scientists first encountered diseases caused by these proteins, the reaction was disbelief. The symptoms were real and terrifying, but no virus or bacterium could be found. Brains showed damage that looked like slow erosion, full of holes, as if reality itself had begun to rot. The idea that a protein alone could cause this kind of destruction was met with resistance. It sounded like superstition, wrapped in science, but the evidence kept piling up. Experiments repeated the same result. Something that should not work was working. The existence of zombie proteins forces biology to admit a quiet truth. Life is not only controlled by living things. Shape matters. Structure matters. Sometimes the difference between health and collapse is not what something is made of, but how it is folded. That realization makes the world feel more fragile. It means that the line between order and chaos can be crossed without life, without intention, without warning. In the grand scheme of things, zombie proteins remind us that nature does not promise safety. It only promises consistency, and even that can bend. The systems keeping us alive are elegant, but they are also vulnerable to strange loopholes. As you drift towards sleep, it is worth sitting with that thought. The world is not only stranger than we imagine, it is stranger than we are comfortable admitting, and sometimes the most unsettling forces are not alive at all. Viroids. There are moments when learning about biology feels comforting, as if life follows a set of quiet rules that keep everything stable and predictable. Cells have boundaries, organisms need energy, and survival usually comes with complexity. But every now and then, something appears that does not just bend those rules, it ignores them entirely. Viroids belong to that category. They matter because they quietly challenge our deepest assumptions about what life needs in order to exist, persist, and cause harm, reminding us that nature does not always respect the frameworks we build to feel safe and certain. A viroid is a tiny piece of genetic material that can infect plants and cause disease, even though it is not a living organism in the traditional sense. What makes viroids feel so unsettling is how little they are. They are made only of ribonucleic acid, with no protective shell, no proteins, no machinery, and no apparent tools to survive on their own. They do not move, they do not eat, and they do not reproduce by themselves. And yet, when they enter a living plant cell, they somehow manage to hijack the system and cause real damage. Leaves curl, fruits shrink, growth becomes distorted, and entire crops can fail. It feels wrong. We expect danger to come from something complex, something active, something alive. But viroids are almost nothing, and that is what makes them so disturbing. According to the basic rules of biology, this system should not exist at all. For decades, scientists believed that to replicate, something must carry instructions and the tools to carry them out. Viruses already stretched that idea by relying on host cells, but even viruses have protein coats and carefully structured components. Viroids have none of that. They are just naked genetic information, no proteins to protect them, no enzymes to help them copy themselves. By all logic, they should be harmless fragments, broken leftovers of real organisms. Biology taught us that without structure and function working together, survival is impossible. Viroids seem to violate that lesson entirely. When scientists first encountered viroids in the 20th century, the reaction was not excitement, but confusion. Plant diseases were appearing that could not be explained by bacteria, fungi, or viruses. Researchers kept looking for missing pieces, assuming they had overlooked a protein or a hidden structure. But the more carefully they studied these agents, the more uncomfortable the truth became. There was nothing else there. What they had discovered was a system that worked precisely because it was simple, not despite it. Many struggled to accept that something so minimal could behave with such effectiveness. It felt like discovering a sentence that could rewrite itself without a pen. The implications of viroids are quietly profound. They suggest that life and non-life are not separated by a clean boundary, but by a blurry gradient. They hint that replication, influence, and harm do not require intention or awareness. Viroids remind us that biology is not always about building upward into complexity, but sometimes about stripping down to the bare minimum and still functioning. In the larger picture, This forces us to rethink how life might begin, how it might exist elsewhere, and how fragile the systems we depend on truly are. The world is not just shaped by grand organisms and visible forces, but also by invisible strings of information, drifting silently, waiting for the right moment to awaken chaos. And once you know they exist, it becomes harder to believe that nature is as orderly as it appears in the daylight. Pandoravirus. There is something quietly unsettling about Pandoravirus, not because it looks frightening, but because it feels like a mistake that slipped past the rules of nature. It is massive where it should be tiny, complex where it should be simple, and strangely independent for something that is not even supposed to count as fully alive. Pandoravirus carries an overwhelming amount of information, packed into a structure that does not behave the way we expect a virus to behave. It does not rush. It does not hurry to explain itself. It simply exists, calm and indifferent, as if daring biology to explain why it is here at all. Pandoravirus is a giant virus that infects single-celled organisms and carries an enormous amount of genetic instructions, blurring the line between viruses and living cells. What makes Pandoravirus feel like an anomaly is how wrong it seems at first glance. Most viruses are tiny and simple, stripped down to the bare minimum needed to survive. Pandoravirus is the opposite. It is larger than many bacteria and contains more genetic instructions than some organisms we comfortably call alive. Even more unsettling is that a large portion of its genetic material appears to have no known purpose. Scientists look at these instructions and cannot match them to anything seen before. It is as if nature wrote an entire book in a language no one recognizes, then quietly slipped it into a virus. This makes Pandoravirus feel less like a variation and more like an intruder from a different set of rules. According to what we normally believe about biology, something like Pandoravirus should not exist at all. Viruses are expected to be simple parasites, completely dependent on the cells they infect, carrying only the instructions they absolutely need. Complexity is supposed to belong to living cells, not to viruses. There is also a long-held idea that life becomes more complex in slow, traceable steps. Pandoravirus seems to ignore this expectation. It arrives already complicated, already bloated with information, without a clear evolutionary story that makes sense. By normal standards, it is too complex to be a virus and too dependent to be a living cell, leaving it stranded in an uncomfortable middle ground. When scientists first encountered Pandoravirus in the early years of the 21st century, confusion came before understanding. At first, it was not even recognized as a virus. Its size and structure led researchers to assume it was some kind of cell. Only later did experiments reveal that it behaved like a virus, hijacking other cells to reproduce. This realization was met with disbelief. Some scientists resisted the idea entirely, arguing that such a thing should not qualify as a virus. The discovery forced a slow and uneasy acceptance that the categories used to describe life were not as solid as once believed. The existence of Pandoravirus carries unsettling implications. It suggests that life is not neatly divided into boxes, but instead spills across boundaries we invented for comfort. It hints that there may be other forms of biological organization waiting to be discovered, forms that do not fit our definitions and do not care to. Pandoravirus reminds us that nature is not obligated to make sense to us. It operates on a deeper logic, one that allows contradictions to sit quietly in plain sight. As you reflect on this, it becomes easier to feel that life is not a clean story with clear chapters, but a vast and unfinished manuscript, filled with passages we have not yet learned how to read. Gene transfer agents, GTAS. There are things in biology that feel orderly and sensible, like a quiet agreement everyone follows. Cells protect themselves. Life struggles to survive. Information is guarded, passed on carefully, almost jealously, from one generation to the next. But hidden deep in the microscopic world is a biological system that behaves differently. It feels generous in a way that borders on self-destruction. It breaks things apart not to grow stronger, but to scatter pieces of itself into the world, hoping something else might use them. It is subtle, almost invisible, yet deeply unsettling once you realize what it is doing. Gene transfer. Agents are tiny particles made by certain bacteria that package random pieces of their own genetic material and release them into the environment for other bacteria to take in. At first glance, this feels deeply wrong. These particles look like viruses, but they are not trying to infect anything for their own benefit. They cannot reproduce. They do not carry instructions to make more of themselves. Instead, they act like biological messengers that tear fragments out of a living cell and give them away freely. Even stranger, the bacteria that create them usually die in the process. A living organism willingly destroys itself just to send pieces of its own genetic code into the world, with no guarantee that those pieces will help anything related to it. It feels less like survival and more like sacrifice. This is what makes gene transfer agents feel like an anomaly. Biology teaches us that life is shaped by survival. Traits that help an organism live and reproduce tend to remain. Traits that kill the organism tend to disappear. Yet here is a system where cells actively produce structures that lead to their own death. The genes being shared are random, not carefully selected. There is no targeting, no planning, no clear reward. It feels like nature allowing a mistake to persist, as if the rules were briefly ignored and never corrected. Under normal biological thinking, this system should not exist at all. Scientists expected genetic information to be tightly controlled, passed only to offspring or occasionally stolen by viruses acting in their own interest. A system where an organism builds complex machinery to give away its own genes at the cost of its own life makes no sense under simple survival logic. There is no immediate benefit to the individual cell. There is no clear payoff. According to classic rules, such a trait should be removed over time. Yet it remains, quietly operating in microbial communities across the planet. When scientists first began to notice gene transfer agents, confusion followed. At first, they were mistaken for broken viruses or laboratory contamination. The idea that cells were intentionally producing virus, like particles without any ability to reproduce, seemed absurd. Researchers struggled to explain why such a system would be preserved. Resistance was not loud, but it was real. The system worked too consistently to be ignored, yet too irrational to fit existing explanations. Something was happening that biology did not have comfortable language for. The implications of this are quietly profound. Gene transfer agents suggest that life does not always operate at the level of the individual. Sometimes survival happens at the level of the community, or even at the level of information itself. Genes may care less about the body they live in, and more about continuing in any form possible. This system hints that cooperation and sacrifice are not rare accidents, but deeply embedded strategies. It reminds us that life is not always efficient, not always fair, and not always understandable. In the presence of gene transfer agents, biology feels less like a machine and more like a conversation. Information flows freely. Boundaries blur. Survival becomes collective, fragile, and strangely generous. And once you notice that, the living world no longer feels solid and predictable. It feels like something held together by quiet agreements we are only beginning to overhear. Immortal jellyfish. There is something quietly unsettling about a creature that refuses to follow the basic rhythm of life. While almost everything around us moves forward in one direction, from beginning to growth to decline, this small and fragile being seems to hesitate, pause, and then simply turn back. It drifts through the ocean without urgency, translucent and delicate, shaped like a soft bell with gentle trailing arms. It looks harmless, almost forgettable, yet hidden within it is a trick that feels less like biology and more like a mistake in the rules themselves. The immortal jellyfish is a tiny sea creature that can restart its own life cycle and return to a younger state instead of dying. What makes this creature feel like an anomaly is not its appearance, but its behavior when things go wrong. When most living beings are injured, starved, or reach the end of their lifespan, their story ends there. The immortal jellyfish does something else entirely. When stressed or damaged, it does not push forward toward death. Instead, it folds inward, sinks to the ocean floor, and begins to undo itself. Its adult body dissolves into a simpler form, and from that simplicity, it slowly rebuilds itself as if time has been reversed. At first glance, this feels deeply wrong. It is as if nature briefly forgot that aging is supposed to be permanent. According to normal biological rules, this should not be possible at all. Living things are built to move in one direction only. Cells specialize, take on specific roles, and lose the flexibility they once had. Once a cell becomes part of muscle, skin, or nerve tissue, it is expected to stay that way until it dies. Aging is considered unavoidable, a consequence of wear, damage, and time itself. Death is not optional in this framework. Yet the immortal jellyfish seems to ignore this expectation, quietly stepping backward across a line that everything else respects. Scientists first encountered this strange behavior not as a bold discovery, but as a confusing observation. Jellyfish kept in laboratory tanks were expected to die after reproducing, just like their relatives. Instead, some of them appeared to vanish, only for new juvenile jellyfish to appear later in the same container. At first, this was treated as a mistake or contamination. It took time for researchers to accept that the same individual was effectively starting over. The idea was uncomfortable. It suggested that something everyone assumed to be fixed was not as solid as believed. The implications of this are both fascinating and humbling. The immortal jellyfish does not mean that life has solved death, nor does it promise eternal youth for other creatures. It still faces predators, disease, and accidents. But its existence quietly reshapes how we think about limits. It shows that aging is not a single universal rule, but a strategy that life often uses and sometimes abandons. Somewhere in the ocean, a creature is demonstrating that the story of life can loop, bend, and rewrite itself. In the grand scheme of things, this reminds us that nature is not obligated to make sense to us. Beneath calm surfaces and familiar patterns, there are systems that challenge our confidence and invite a softer kind of curiosity. The world is not just stranger than we expect. It may be stranger than we are even capable of fully understanding, patiently waiting for us to notice. Tardigrade Cryptobiosis. There is a tiny creature that lives at the edge of what we think life is allowed to do. It is small enough to hide between grains of sand, slow enough to be overlooked, and unremarkable at first glance. Yet it carries a quiet defiance in its body. It can dry out completely, freeze solid, burn under extreme heat, survive crushing pressure, and endure the vacuum of space. It does not scream its resilience. It simply waits, patient, still, almost as if time itself has lost interest in it. Tardigrade cryptobiosis is a biological state where a tiny animal shuts down nearly all signs of life and waits in a suspended condition until the world becomes safe again. What makes this system feel so wrong is how absolute it is. When a tardigrade enters cryptobiosis, it loses almost all of the water inside its body. Its metabolism slows to the point that it is nearly undetectable. There is no movement, no growth, no repair. For all practical purposes, it looks dead. Not mostly dead. Not barely alive. Just gone. And yet, years later, sometimes decades later, when water returns, it unfolds itself and continues living as if nothing happened. No memory loss, no missing pieces, no gradual restart. It simply resumes. This feels unsettling, because life, as we understand it, is supposed to be fragile. Cells rely on water to keep their structure. Proteins collapse when dried. DNA breaks under radiation. Heat denatures the machinery of life. Freezing shreds cells from the inside. According to everything we are taught, removing water and stopping metabolism should mean irreversible death. There should be no coming back from that. Life is supposed to be a process, not a pause button. Under normal biological rules, cryptobiosis should not exist at all. Living systems are defined by constant activity, breathing, repairing, consuming energy, maintaining balance. Stop those processes, and the system falls apart. Cells accumulate damage. Chemical reactions drift into chaos. Entropy wins. The idea that an animal could stop all of this, remain intact, and then restart later violates the expectation that life must be continuous. It challenges the belief that death is a clear boundary rather than a temporary condition. When scientists first began to study tardigrades seriously, the reaction was confusion mixed with disbelief. Early observations were often dismissed as mistakes or exaggerations. Surely these animals had not truly dried out completely. Surely they were not really frozen solid. Surely something was still quietly running inside them. But repeated experiments removed water entirely, exposed them to extremes, and still they returned. Again and again. The more carefully researchers tested them, the more uncomfortable the result became. These creatures were doing exactly what biology said they could not. The existence of cryptobiosis forces a quiet reconsideration of what life actually is. It suggests that life may not be defined by constant activity, but by the potential to resume activity. It hints that survival is not always about strength or speed, but about patience, about waiting without breaking. In a larger sense, it reminds us that nature does not follow our rules. We observe patterns and call them laws, but occasionally something slips through the cracks and survives anyway. In the presence of tardigrade cryptobiosis, the world feels less solid. Death looks less final. Life feels more like a spectrum than a switch, and somewhere, in a grain of dust, a tiny creature remains folded in on itself, neither alive nor gone, quietly proving that reality is far stranger than it appears. HeLa cells. There is something quietly unsettling about a living thing that refuses to behave. Something that grows when it should stop, survives when it should fade, and keeps going long after the moment it was meant to end. Most living systems are defined by limits. They age, they weaken, they eventually fail. This system does none of that. It is persistent, tireless, and strangely indifferent to time itself, as if it slipped past a rule that everything else must obey. HeLa cells are human cells taken from one woman that continue to live and divide endlessly in laboratories around the world. At first glance, this already sounds wrong. Human cells are not supposed to live forever. Every cell in your body carries a kind of internal clock. With each division, it moves closer to a final point where it can no longer copy itself safely. This limit protects the body. It prevents chaos. It ensures that growth stays controlled and purposeful. But HeLa cells ignore that clock completely. They divide again and again without slowing down, without aging, without asking permission. They behave more like a spreading idea than a piece of flesh. What makes this feel so unsettling is how aggressively alive they are. In the right conditions, they multiply faster than normal human cells. They crowd out other cells. They adapt quickly. They survive freezing, shipping, and environments that would kill most living tissue. In some laboratories, they grew so fast that they accidentally contaminated other experiments, quietly replacing the original cells without anyone noticing. It was as if the cells wanted to exist more than they wanted to belong. According to normal biological rules, this should not happen. Cells are meant to be temporary workers, not eternal beings. When they divide too much, errors build up. DNA becomes unstable. Systems fail. The cell is supposed to stop or self-destruct before it causes harm. This is one of the most basic safety rules of life. HeLa cells break that rule completely. They continue dividing even when their internal instructions are damaged. Instead of collapsing, they thrive. By all expectations, they should have burned out quickly. Instead, they became functionally immortal. When scientists first encountered these cells in the early 1950s, the reaction was disbelief. Cells taken from a human tumor were expected to die within days, like all the others before them. But these did not. Days passed, then weeks, then months. The cells kept growing. Researchers shared them with other laboratories, expecting the effect to fail somewhere along the way. It never did. What should have been impossible kept happening in front of their eyes. Something was deeply wrong with the rules they thought they understood. The implications are both extraordinary and uncomfortable. HeLa cells helped scientists develop vaccines, study cancer, understand genetics, and explore the basic mechanics of life. They have been involved in tens of thousands of experiments, yet they also force a difficult realization. Immortality at the cellular level is not a gift. It is unstable. It is invasive. It comes from something broken rather than perfected. HeLa cells remind us that nature does not always build with elegance. Sometimes it stumbles into strange solutions that work too well. Life, it turns out, is not just fragile and temporary. Under the right conditions, it can become relentless, quietly unstoppable. And that makes the world feel a little less predictable, a little less safe, and far more mysterious than we like to believe when we turn out the lights and close our eyes. Bacterial Endospores. Some biological systems feel elegant and alive in the way we expect life to be. They breathe, they grow, they react, they change. But every once in a while, biology presents something that feels almost mechanical, as if it stepped out of a different set of rules entirely. Something that does not behave like life should. Something that waits instead of lives. Bacterial endospores belong to this strange category. They are quiet, patient, nearly indestructible, and unsettling in how little they resemble the fragile living things we think we understand. In the simplest possible terms, a bacterial endospore is a hardened survival form that some bacteria create to endure conditions that would normally kill life. What makes this system feel wrong at first glance is how completely it shuts down. Most living things survive by adapting while still being alive. They slow down, they conserve energy, they find shelter. Endospores do something far more extreme. They stop all visible activity. No growth, no movement, no feeding, no reproduction. To the outside world they look dead, and yet, given the right conditions, they come back. Not weakened, not damaged, just alive again, as if time never passed. Some endospores have survived boiling heat, crushing pressure, radiation, deep freezing, toxic chemicals, and years or even centuries of complete dryness. This feels like nature cheating its own rules. According to the basic rules of biology, this should not be possible. Life depends on constant chemical reactions. Cells need water. Proteins break down over time. DNA accumulates damage. Heat scrambles structure. Radiation tears molecules apart. Every biology textbook teaches that life is delicate, temporary, and always decaying. A cell that completely shuts down its chemistry should fall apart, like a clock left for rust. Yet endospores do not decay the way they should. They protect their genetic material, remove most of their water, and lock themselves inside layers so tough they rival engineered materials. By everything we thought we knew, a living thing that does this should cross the line into death. But it does not. When scientists first studied these structures seriously, confusion followed. Samples thought to be sterile suddenly showed growth. Bacteria believed to be destroyed returned without explanation. Researchers resisted the idea at first, assuming contamination or error. The notion that something alive could endure such extremes without metabolism felt absurd. Over time, repeated experiments forced acceptance. These were not mistakes. Nature had quietly built something that behaves like a pause button for life. The existence of bacterial endospores forces an uncomfortable realization. Life is not defined only by activity. It can also be defined by potential, by waiting, by persistence beyond time scales we intuitively understand. Endospores blur the boundary between life and non-life, between survival and suspension. They suggest that life is less fragile than we imagine, but also more alien. If something so small can endure almost anything by simply refusing to participate in the world for a while, then biology is not just about growth and change, it is also about stillness, about endurance, about waiting patiently for the universe to become gentle again. Anglerfish sexual parasitism. In the deepest parts of the ocean, where sunlight never reaches and silence feels permanent, life follows rules that seem written for another universe. Creatures there are shaped by scarcity, pressure, and darkness so complete that even the idea of sight feels optional. Among these organisms, there is one system so strange that it sounds less like biology and more like a quiet nightmare. It is intimate, irreversible, and disturbingly efficient. It is not violent in the way predators are violent, but unsettling in a slower, more existential way. This system forces us to rethink what it even means to be an individual living thing. Anglerfish sexual parasitism is a reproductive system where a tiny male permanently fuses his body to a much larger female and survives only as part of her. At first glance, this system feels wrong. The male anglerfish does not court, compete, or even remain independent. Instead, he bites the female and never lets go. Over time, his body merges with hers, His organs shrink, his eyes fade, his digestive system disappears. Eventually, he becomes little more than a living attachment whose only remaining function is to provide reproductive cells when needed. He does not leave. He does not grow old on his own. He does not exist as a separate animal anymore. Nature, which we often imagine as valuing survival and independence, allows one life to slowly erase itself inside another. What makes this feel especially disturbing is how final it is. There is no separation, no reversal, no second chance. The male sacrifices everything that makes him a complete organism. From a human perspective, it feels like a violation of identity itself. We are used to thinking of reproduction as cooperation between two whole individuals. Here, one individual gives up being whole at all. It feels like something biology should reject outright, as if it breaks an unspoken rule about life needing boundaries. According to normal biological expectations, this system should not exist. Living creatures are supposed to maintain their own bodies, their own immune systems, and their own internal balance. When foreign tissue enters another body, the immune system attacks it. That is one of the most basic rules of biology. Survival depends on recognizing what is self and what is not. By that logic, a male anglerfish attaching to a female should trigger rejection, infection, or death. Two separate organisms should not be able to merge their blood and tissues without catastrophic consequences. Yet here, not only does it happen, it works perfectly. When scientists first began to understand this system, confusion came before acceptance. Early observations were dismissed or misunderstood. Some thought the fused males were parasites or deformities. Others assumed the males were a different species entirely. The idea that a vertebrate animal could permanently fuse with another without immune rejection seemed impossible. It took repeated observations and careful study for researchers to accept that this was not an accident or disease, but a deliberate and successful strategy shaped by evolution. What should not work was working quietly and reliably in the dark. The existence of this system changes how we think about life itself. It shows that individuality is not as fixed as we imagine. That survival does not always mean independence. In an environment where finding a mate is nearly impossible, nature chose permanence over freedom. Anglerfish sexual parasitism reminds us that biology is not designed to be comforting or intuitive. It is designed to persist. And sometimes, persistence looks like something we would never allow, never choose, and never fully understand. The deeper we look, the more we realize that the world is not built around our assumptions. Life is stranger than logic. Identity is more flexible than comfort allows. And in the quiet darkness beneath the ocean, nature continues to solve problems in ways that feel less like answers and more like unsettling truths we are still learning how to sit with. Devil facial tumor disease. There are biological systems that feel elegant and balanced, and then there are those that feel almost wrong, as if they slipped through a crack in the rules of life. Devil facial tumor disease belongs to that second category. It is quiet, persistent, and disturbingly effective. It does not rely on clever chemistry or complex cooperation between cells. Instead, it survives by breaking expectations in the most unsettling way possible. It turns the body itself into a vehicle, transforming something meant to protect life into something that spreads destruction. What makes it unique is not just that it is deadly, but that it behaves in a way that feels more like a living paradox than a disease. In simple terms, devil facial tumor disease is a contagious cancer that spreads between Tasmanian devils through biting. At first glance, this makes very little sense. Cancer is supposed to be personal. It begins when a cell inside a body goes wrong and starts multiplying without control. It is tragic, but it is contained within one individual. Devil facial tumor disease does not follow that pattern. Instead of staying trapped in one animal, the cancer itself moves from one body to another. When Tasmanian devils bite each other during feeding or mating, living cancer cells pass directly into the wounds. Those cells do not die. They take root. They grow. They form massive tumors on the face and mouth, eventually making it impossible for the animal to eat. It feels unsettling because it blurs the line between disease and organism. The cancer behaves almost like a parasite, yet it is not one in the usual sense. According to the normal rules of biology, this should not be possible. The immune system exists to prevent exactly this kind of invasion. Every body has a biological identity, like a molecular fingerprint. When foreign cells enter, the immune system is supposed to recognize them as intruders and destroy them. This is why organ transplants are so difficult and why blood types matter. A random clump of cells from another animal should be eliminated almost instantly. A cancer from a different body should be even easier to reject. By all expectations, devil facial tumor disease should fail the moment it enters a new host. Yet it does not. It survives. It grows. It ignores rules that seem fundamental to life itself. Scientists were deeply confused when they first realized what they were seeing. At first, it looked like an outbreak caused by a virus or bacteria, but no infectious agent could be found. When researchers examined the tumors more closely, they noticed something shocking. The cancer cells in different animals were genetically almost identical. They did not match their hosts. They matched each other. This meant the disease was not spreading an instruction for cancer, it was spreading the cancer itself. Many scientists struggled to accept this idea because it challenged a basic assumption about how bodies defend themselves. The realization felt less like solving a puzzle and more like discovering that a wall everyone trusted had never really been there. The existence of devil facial tumor disease changes how we think about biology in a quiet but profound way. It reminds us that the immune system is powerful, but not perfect. It shows that under certain conditions, even the most basic safeguards can fail. It also reveals how fragile populations can be when genetic diversity is low, allowing something so improbable to take hold. On a larger scale, it suggests that life is more experimental than we like to believe. Nature does not always follow clean rules. Sometimes it finds paths that feel disturbing, even unfair. And once you notice that, it becomes harder to see the living world as stable or predictable. Instead, it feels like something constantly balancing on the edge of possibility, where even the impossible can quietly learn how to survive. Sacculina. There are biological systems that impress us because they are complex, efficient, or beautifully balanced. And then there are systems that unsettle us because they seem to erase the very idea of balance altogether. Sacculina belongs to the second category. It is not impressive in a heroic way. It does not build, protect, or cooperate. Instead, it quietly dissolves boundaries we assume are fundamental. It rewrites identity, purpose, and even gender with no regard for the rules we expect life to follow. What makes it unique is not how strong it is, but how completely it takes over another living being without ever looking like it should be able to. Sacculina is a parasitic organism that infects crabs and turns them into living extensions of itself, controlling their bodies while erasing its own. At first glance, this system feels deeply wrong. Sacculina begins life as a tiny larva drifting through the ocean, no different from countless others. But when it encounters a crab, it does something that feels almost impossible. Instead of attaching to the outside or living in the gut, it injects itself directly into the crab's body. Once inside, it spreads out like a living network of roots, wrapping around the crab's organs and nervous system. Over time, the crab stops behaving like a crab. It stops reproducing. It stops molting normally. It even stops caring about its own survival in the way it should. The parasite does not just steal nutrients, it rewrites behavior. The crab becomes a caretaker for something that is slowly replacing us from the inside. What makes this feel like an anomaly is how completely Sacculina abandons what we think an animal needs to be. As it matures, it loses its legs, its shell, and nearly all recognizable body parts. It becomes a sack of tissue with one purpose only, and yet somehow it survives. Even stranger, male crabs infected by Sacculina begin to act like female crabs. They develop the instinct to protect and nurture the parasite's reproductive body, gently cleaning it and defending it as if it were their own offspring. Nature, which we expect to reward reproduction and self-preservation, allows a system where an organism destroys both and still thrives. According to normal biological rules, this system should not exist at all. Animals are supposed to have organs, boundaries, and independence. Parasites usually weaken their hosts, but do not completely replace their identity. Hormones and behavior are expected to follow the organism's own genetic instructions. Sacculina violates all of this. It has no clear body. It hijacks another organism's hormones. It forces behavior that goes directly against the host's ability to reproduce or pass on its genes. From a basic survival perspective, it looks like a dead end. Yet it works, again and again, across generations. When scientists first studied Sacculina, they struggled to even classify it. It did not look like an animal. It did not behave like one. Early observers thought it might be a disease or a strange growth rather than a living creature. The idea that a barnacle, something usually stuck harmlessly to rocks, could do this to a crab felt absurd. There was disbelief, confusion, and a quiet discomfort in realizing that something so simple could exert such total control. It was not clever in a human sense. It was simply effective. The existence of Sacculina forces us to rethink what life is allowed to be. It suggests that evolution does not care about fairness, dignity, or even identity. It only cares about persistence. Life can succeed not just by building complexity, but by erasing it. Sacculina reminds us that nature is not always nurturing. Sometimes it is invasive, subtle, and indifferent. And if a system like this can exist, quietly thriving beneath the surface of the ocean, then perhaps the world is far stranger and less stable than we like to believe. Not broken, but unconcerned with our expectations. Kleptoplasty. There are moments in nature that feel quiet and elegant at first, almost gentle, until you realize how deeply wrong they seem. Kleptoplasty is one of those moments. It has a delicate beauty to it, something subtle and almost poetic, as if life itself decided to bend its own rules without asking permission. It is not loud or aggressive. It does not announce itself as a rebellion. Instead, it exists calmly, hidden in shallow waters and soft green hues, doing something that should not be possible, and doing it without apology. Kleptoplasty is the process where certain animals steal parts of plants and use them to make energy from sunlight. At first glance, this feels harmless, even clever. But the more you sit with it, the stranger it becomes. In our basic understanding of life, plants and animals live very different roles. Plants capture sunlight. Animals eat plants or other animals. There is a clear boundary, a quiet agreement that has shaped biology textbooks and classrooms for generations. Kleptoplasty steps over that boundary without hesitation. Some animals, most famously a small sea slug, eat algae and somehow keep the algae internal machinery alive inside their own bodies. These stolen parts continue to absorb sunlight and produce energy, not for the plant, but for the animal that took them. What makes this unsettling is not just that it happens, but that it works. The animal does not digest these parts like normal food. It keeps them. It maintains them. It benefits from them. This feels like a glitch in the system, like an animal borrowing a feature it was never meant to have. It is as if a cow suddenly decided to grow leaves and calmly photosynthesize in a field, and no one could explain why it did not collapse under its own contradictions. According to the usual rules of biology, this should not exist at all. The tiny structures that capture sunlight are delicate and require constant support from plant genes. They are supposed to break down quickly outside of a plant cell. Animals do not have the instructions to maintain them. They lack the tools, the maintenance manuals, the quiet cellular routines needed to keep these systems running. Scientists expected that if an animal swallowed these structures, they would fail almost immediately. Instead, in kleptoplasty, they persist for weeks or even months, functioning inside a completely foreign environment. When researchers first observed this, the reaction was confusion mixed with disbelief. Early observations were often dismissed as misunderstandings or short-lived accidents. Surely the animal was just digesting the algae slowly. Surely the green color was cosmetic. But repeated observations refused to align with those explanations. Experiments showed that these animals could survive longer in sunlight, even when deprived of food. Something was clearly happening that should not have been happening. It forced scientists to admit that the boundary between plant and animal was not as rigid as once believed. The existence of kleptoplasty quietly reshapes how we think about life itself. It suggests that evolution is not always clean or well organized. Sometimes it is opportunistic, messy, and surprisingly flexible. Life does not always respect the categories we create for it. Instead, it borrows, adapts, and experiments in ways that feel almost uncomfortable to acknowledge. If an animal can temporarily become plant-like, then the lines we draw between living things begin to blur. Kleptoplasty reminds us that nature is not obligated to make sense to us. Beneath the calm surface of the world, there are systems quietly operating that defy expectation, surviving in ways that feel fragile and miraculous at the same time. It leaves us with a gentle unease and a deeper wonder, as if the living world is whispering that we have only understood a fraction of what it is capable of becoming. Apoptosis. There is something quietly unsettling about a system that is built not to protect life, but to end it from the inside. Apoptosis is precise, deliberate, and almost polite in the way it works. It does not involve chaos or violence. It does not spread damage to neighboring cells. Instead, it follows a careful internal script, as if each cell carries a final instruction written long before it was ever needed. This system exists in nearly every complex living organism, working constantly and invisibly, shaping tissues, sculpting organs, and removing parts of us without our awareness. It is one of the few systems in biology that feels intentional in a way that borders on philosophical. In simple terms, Apoptosis is the process by which a living cell intentionally shuts itself down and dies for the benefit of the whole body. At first glance, this feels deeply wrong. We tend to think of life as something that fights to survive at all costs. Every biology lesson reinforces the same idea. Living things compete, adapt, repair, and resist death. Yet apoptosis asks cells to do the opposite. It asks them to recognize when they are no longer needed, or when they might become dangerous, and then calmly remove themselves. No external attack. No injury. Just an internal decision to stop existing. It feels like a betrayal of the most basic rule of life. What makes this even more unsettling is how routine it is. Apoptosis happens during early development to separate fingers and toes. It happens every day to remove cells that are old, damaged, or slightly incorrect. Entire populations of cells are quietly erased to keep tissues balanced. From the outside, nothing looks wrong. The body appears stable, but beneath the surface, countless tiny deaths are happening on schedule. Nature, it seems, did not just allow this. It depends on it. According to what we once believed about biology, this system should not exist at all. Cells were expected to act like simple survival machines. Their goal was assumed to be persistence: divide, repair, and continue. Death was thought to come only from external harm, like injury, starvation, or infection. The idea that a cell would carry a built-in self-destruction program seemed wasteful, even illogical. Why would evolution favor something that actively kills its own successful units? Why would a healthy cell ever cooperate with its own disappearance? When scientists first began to observe this behavior, it caused confusion and resistance. Under the microscope, cells were seen shrinking, fragmenting, and being neatly cleared away without any sign of damage. At first, this was dismissed as an artifact or a mistake in observation. Many believed these cells were simply dying accidentally. But over time, patterns emerged. The same sequence of events repeated. The same signals appeared. The same orderly ending unfolded again and again. It became impossible to ignore that this was not failure. It was design. The realization forced a quiet shift in how life itself was understood. Survival was no longer the only goal. Cooperation mattered more. A cell that refuses to die when it should can become cancerous. A cell that dies at the wrong time can cause degeneration. Life, it turns out, is balanced on a thin edge between persistence and surrender. Apoptosis is the mechanism that walks that edge every moment. The existence of apoptosis suggests that life is not built around endless continuation, but around controlled endings. It tells us that death is not always an enemy of life, but sometimes its caretaker. Without this system, bodies would grow misshapen, unstable, and eventually unlivable. What feels impossible at first glance becomes essential when viewed as part of a larger whole. And perhaps that is the quiet lesson hidden inside this system. That survival is not about clinging forever, but about knowing when to let go. That complexity requires loss. That order sometimes depends on disappearance. Apoptosis reminds us that beneath the surface of living things, there is a constant negotiation between existence and absence, happening so smoothly that we never feel it. The world is not just alive. It is carefully, deliberately unfinished. Retrotransposons. There is something quietly unsettling about the idea that not everything inside your body is working for you. We often imagine biology as careful and precise, every part serving a clear purpose, every instruction written with intention. Yet hidden deep inside your cells is a system that feels almost rebellious. It copies itself, moves without asking permission, and leaves traces behind like footprints in wet cement. It is ancient, persistent, and strangely creative. It does not announce itself, but it has shaped who you are more than most people realize. Retrotransposons are pieces of genetic material inside living cells that can copy themselves and insert those copies into new places within the genome. At first glance, this feels like a mistake. Your genome is supposed to be a carefully guarded instruction manual, telling your cells how to grow, repair, and survive. Random copying and pasting inside that manual sounds dangerous, like someone scribbling new sentences into a book while you are still trying to read it. Retrotransposons do exactly that. They make a copy of themselves through an indirect route and then slip that copy back into the genetic text, sometimes landing in places that matter. They do not ask whether the spot is safe. They do not check if they will disrupt something important. They simply move. What makes this especially strange is that these elements behave a bit like viruses, but they are not invaders from the outside. They are part of us. Some of them make up a surprisingly large portion of human genetic material. They are repeated again and again, scattered across the genome like echoes of a forgotten message. From a distance, this looks like chaos. Why would life tolerate something that copies itself for no clear reason and risks breaking vital instructions along the way? According to the usual rules of biology, this should not be allowed. Harmful changes are supposed to be removed over time. Systems that waste energy or threaten survival are expected to disappear through evolution. Stability is valued, precision is rewarded. Yet retrotransposons do the opposite. They introduce uncertainty. They create variation that can be harmful, neutral, or occasionally useful. By all logic, a self-copying genetic element that can disrupt important functions should have been eliminated long ago. The expectation was that genomes would be streamlined and efficient, not cluttered with wandering sequences that seemed to serve no obvious purpose. When scientists first began to understand what was happening, the reaction was not immediate acceptance. There was confusion and skepticism. The idea that genes could move around inside the genome sounded almost heretical. Genes were thought to be fixed, orderly, and stable. The discovery that parts of the genome could rearrange themselves felt like finding moving walls inside a house you thought was solid. It took time for researchers to accept that this was not an error in observation, but a real and widespread feature of life. As studies continued, it became clear that retrotransposons were not rare accidents. They were everywhere. Plants, animals, and even humans carried them. Some were active, others silent, like fossils from an earlier age. Slowly, disbelief turned into uneasy curiosity. If this system was so dangerous, why had life kept it around for so long? The implications are quietly profound. Retrotransposons remind us that biology is not perfectly engineered. It is patched together, shaped by trial and error, and full of compromises. The same system that can cause disease or genetic instability can also drive evolution and creativity. By reshuffling genetic material, retrotransposons have helped create new genes, new traits, and new possibilities. They blur the line between damage and innovation. In the end, their existence suggests that life is not as clean or predictable as we like to believe. It grows by allowing a certain amount of mess, a certain amount of risk. Deep inside, every cell is a reminder that order and disorder are not enemies, but partners. And as you sit with that thought, it becomes harder to see life as fragile clockwork, and easier to see it as something stranger, more resilient, and quietly mysterious. Eusociality. There is something quietly unsettling about a society that runs with perfect order and almost no individuality. A place where most lives are never meant to shine, never meant to reproduce, and never meant to matter on their own. Everything moves with calm precision, as if guided by a single shared purpose. No one asks why they exist. No one chooses their role. Some are born to rule, some are born to work, and some are born only to die for others. It feels less like a community and more like a living machine, breathing and growing without hesitation or doubt. Eusociality is a way of life where a group of animals lives together in extreme cooperation, with only a few individuals reproducing, while the rest spend their entire lives serving the group. At first glance, this makes no sense at all. Most members of a eusocial group never pass on their own genetic material. They work endlessly, gather food, defend the nest, and care for young that are not theirs. Many of them will sacrifice their lives without resistance or hesitation. From a human point of view, it feels deeply wrong. Nature, as we usually understand it, rewards survival and reproduction. Yet here is a system where survival is optional and reproduction is forbidden for most. It feels like something biology should have rejected immediately. According to basic biological rules, this system should not exist. Life is supposed to favor individuals that reproduce the most. Traits that help an individual survive and have offspring are the ones that continue. Under this logic, an organism that never reproduces should be a dead end. It should disappear over time, erased by evolution. Eusociality appears to break this rule completely. Why would nature allow a body to exist only as a tool for others? Why would a living being evolve to serve instead of reproduce? By normal expectations, these creatures should have been outcompeted and wiped out long ago. When scientists first seriously studied this behavior, it caused confusion and discomfort. The idea that evolution could favor self-sacrifice over self-interest felt almost heretical. Early explanations were resisted because they seemed to undermine the very foundation of natural selection. Researchers struggled to explain how a system built on cooperation could survive in a world driven by competition. Some dismissed it as a strange exception. Others suspected there was something wrong with the theory itself. It took time for scientists to accept that the rules might be more flexible and more unsettling than they had assumed. Eventually, it became clear that eusociality was not a mistake. It was not a loophole. It was a different way for life to succeed. Instead of the individual being the unit of survival, the group became the unit. The colony acted like a single organism, with different bodies performing different functions. In this light, the workers were not failures. They were organs. They were hands and immune systems and muscles, all serving something larger than themselves. The existence of eusociality changes how we think about life. It suggests that individuality is not as sacred as we believe, that survival does not always belong to the strongest or the smartest, but sometimes to the most cooperative. It hints that life is willing to erase the self entirely if it means the system endures. In the quiet order of these societies, we see a reminder that nature does not care about meaning, fairness, or freedom. It only cares that something continues. And perhaps that is what makes eusociality so haunting. It shows us that life can thrive without choice, without identity, and without mercy. It leaves us wondering how many other rules we assume are fixed, when in reality they are only suggestions waiting to be quietly ignored. Bdelloid Rotifers. There is something quietly unsettling about life forms that refuse to follow the rules we think are unbreakable. Bdelloid Rotifers belong to that category. They are tiny, almost invisible creatures, living in places most people never notice, hiding in droplets of water, damp moss, or thin films of moisture on leaves and stones. They survive freezing, drying, radiation, and time itself with a calm indifference. They do not rush, they do not struggle, they simply persist. While most living things depend on constant balance and delicate conditions, these creatures seem to pause their existence, wait patiently, and then return as if nothing happened. Their very existence feels like a quiet challenge to what we assume life must be. Bdelloid rotifers are microscopic animals that live in water and reproduce for millions of years without ever needing males. What makes this system feel so strange is not just their size or where they live, but how they live. Bdelloid rotifers reproduce entirely on their own. Every individual is female. There is no mating. There is no exchange of traits between two parents. Each new rotifer is essentially a copy of the one before it. At first glance, this might not sound shocking, but when you consider how life usually works, it starts to feel deeply wrong. Variety is supposed to be essential. Mixing traits is supposed to protect species from disease and extinction. Yet these creatures have gone on like this for an almost unimaginable stretch of time, surviving disasters that wiped out far more complex life. According to normal biological rules, this system should collapse quickly. When a species reproduces without mixing traits, harmful changes build up over generations. Diseases should spread easily. The environment should eventually outpace their ability to adapt. In simple terms, life without variety is expected to weaken, decay, and disappear. Scientists once believed that a species like this could not survive long on a geological scale. It was assumed that such creatures would be evolutionary dead ends, brief experiments that nature quickly erased. The shock came when scientists realized that Bdelloid rotifers were not short-lived at all. Fossils and genetic studies showed that they had been living this way for tens of millions of years. That discovery caused quiet disbelief. Researchers searched for hidden males. They assumed something must have been missed. But again and again the evidence pointed to the same conclusion. These animals truly lived without sexual reproduction. Even more unsettling, their genetic material showed signs of borrowing pieces from bacteria, plants, and fungi, almost as if they were patching themselves together from the environment. This was not how animals were supposed to work. The implications of this are subtle but profound. Bdelloid rotifers suggest that life is more flexible than our rules imply. They show that survival does not always require complexity, speed, or even cooperation between individuals. Sometimes persistence comes from stillness, patience, and unconventional solutions. Their existence forces scientists to rethink what is truly essential for life to continue. It reminds us that the frameworks we build to understand nature are temporary, and that reality often operates just outside the edges of our certainty. In the quiet presence of these tiny creatures, there is a gentle message. The world is not only stranger than we imagine, but stranger than we are prepared for. Life finds ways not by obeying expectations, but by slipping through them, and somewhere in a drop of water, a Bdelloid rotifer waits calmly, proving that even the most basic assumptions can be wrong. Syncytium. There are things in biology that feel orderly and predictable, like cells neatly dividing, each one wrapped in its own boundary, each one knowing where it begins and where it ends. We are taught that life is built from these small, self-contained units, separated by membranes, communicating carefully, never losing their individual identity. That picture feels comforting. It suggests control, structure, and rules that keep life from collapsing into chaos. But hidden beneath that tidy image is a biological system that quietly ignores those rules, existing in a way that feels more like a living contradiction than a well-behaved design. A syncytium is a biological structure where many cells merge together and share one continuous body without clear boundaries between them. What makes this system feel unsettling is how completely it abandons the idea of individuality at the cellular level. Instead of separate cells working side by side, a syncytium forms when cell walls dissolve and their contents blend into one large shared space. Multiple nuclei float together inside a single mass, all controlling different regions at once. There is no clear border, no obvious separation, no simple way to say where one cell ends and another begins. At first glance, it looks messy, uncontrolled, and strangely wrong, like a mistake that should have been cleaned up by evolution, but never was. According to the basic rules most of us learn about biology, this should not work at all. Cells are supposed to be sealed off to protect their contents, to regulate what enters and leaves, and to prevent damage from spreading. Without boundaries, problems should cascade instantly. A single error in one area should affect everything. Coordination should collapse. Growth should become unstable. Yet syncytia grow, function, repair themselves, and in some cases perform critical roles in living organisms. They break the expectation that separation is necessary for order. Scientists struggled with this idea when it was first seriously studied. The assumption was that cells without boundaries would be fragile and short-lived, more like biological accidents than functional systems. When researchers observed stable syncytia in muscle tissue, in early development, and even in certain parasites, the reaction was not excitement, but confusion. How could so many nuclei cooperate without constant conflict? How could signals travel accurately without walls to guide them? For a long time, the answer seemed to be that they should not be able to, and yet, under the microscope, they clearly were. What unsettled researchers most was not just that syncytia existed, but that they worked well. Muscle fibers rely on this shared structure to contract smoothly. Early embryos depend on syncytial stages to develop properly. Some organisms use syncytia to grow rapidly and adapt quickly. These systems do not merely survive despite their strange design. They thrive because of it. What once looked like a flaw turned out to be a different strategy entirely. The existence of syncytia quietly reshapes how we think about life. It suggests that boundaries, while useful, are not absolute requirements. Order can emerge without strict separation. Control can exist without walls. Life is not limited to one blueprint, even at its most basic level. In the grand scheme, this means nature is more flexible, more experimental, and more tolerant of strangeness than we often assume. As you sit with this idea, it leaves a gentle unease behind. If something that seems so fundamentally wrong can be essential, then how many other rules are only temporary assumptions? The world may be held together not by rigid laws, but by systems that work simply because they do. And somewhere inside your own body, countless nuclei drift together in quiet cooperation, reminding us that life does not always make sense, and it does not need to. Dictyostelium. Dictyostelium is the kind of biological system that feels quiet and modest at first glance, almost forgettable. It is small, soft, and unimpressive to the naked eye. It lives in soil, feeds on bacteria, and spends most of its existence alone. And yet, hidden inside this simple life is a behavior so strange that it feels almost philosophical. It behaves like a contradiction that learned how to survive. It moves between independence and unity in a way that feels unsettling. as if it cannot decide what kind of life it wants to be. It challenges the idea that living things must follow one clear strategy, one clear identity, or one clear role. Dictyostelium is a type of slime mold that lives as single cells most of the time, but comes together to form one living body when conditions become harsh. What makes this system feel like an anomaly is how casually it breaks the line between being alone and being together. Most of the time, each cell lives its own separate life, eating, moving, and surviving without any leader. But when food runs out, something strange happens. Thousands of these independent cells begin to send signals to one another. They move closer, they gather, they merge into a single crawling mass, as if they suddenly remember they belong to something larger. Even more unsettling, this combined body begins to behave like a single organism, with coordination, direction, and purpose. The strangest part comes next. Once they have gathered, some of the cells willingly sacrifice themselves. They become the stalk of the structure, lifting the remaining cells upward. These remaining cells will survive, disperse, and begin life again. The cells that form the stalk die. They do not reproduce. They gain nothing. They exist only to support others. At first glance, this feels like something nature should never allow. Why would a single cell give up its entire future for neighbors that were strangers just moments ago? According to normal biological rules, this system should not exist at all. Evolution is often explained as a process driven by individual survival and reproduction. Traits that help an individual pass on its genes are favored. Traits that lead to death without reproduction are removed over time. Dictyostelium appears to violate this expectation. These cells are not born into a permanent group. They are not related in a simple family structure. And yet, they cooperate deeply, even to the point of self-destruction. By the usual logic, cheating should take over. Cells that refuse to sacrifice should win. Cooperation like this should collapse. When scientists first began studying Dictyostelium closely, this contradiction caused confusion and debate. Researchers struggled to understand how cooperation could remain stable in such a loose and temporary group. Some expected that hidden rules must exist, or that the system was more selfish than it appeared. Over time, experiments showed that the cooperation was real, measurable, and repeatable. Nature had somehow found a way to balance competition and sacrifice in a setting where it seemed impossible. What looked like a mistake turned out to be a stable strategy. The existence of Dictyostelium quietly reshapes how we think about life. It suggests that cooperation does not always require complex brains, emotions, or moral reasoning. It can emerge from simple rules and shared danger. It reminds us that individuality and unity are not opposites, but states that life can move between when survival demands it. In the grand scheme, this system hints that nature is more flexible than our categories allow. Life does not always choose between selfishness and cooperation. Sometimes it uses both, at different moments, without asking permission. And once you notice that, the world begins to feel less solid, less predictable, and far more mysterious than it seemed before. Gynandromorphs. There is something quietly unsettling about creatures that do not choose a single form. At first glance, they look ordinary, even beautiful, but the longer you look, the more your mind struggles to place them into a clear category. They carry two stories at once, written into their bodies, not as a mistake to be corrected, but as a reality that somehow works. They feel like a pause in nature, a moment where the usual rules hesitate, as if life itself briefly forgot how it was supposed to decide. A gynandromorph is a living organism that develops with both male and female physical traits in the same body. What makes this so strange is how clean the division can be. In some insects and birds, one side of the body can appear male, while the other side appears female, split almost perfectly down the middle. One wing may be colored and shaped differently from the other. One eye may be larger or brighter. Even behavior can seem divided, as if the creature is quietly switching roles depending on which side of itself is acting. This is not a gradual blend or a smooth transition. It looks like two blueprints stitched together, sharing the same heartbeat. To our eyes, it feels wrong in a way that is hard to explain, not ugly or broken, but deeply confusing, like seeing a shadow that does not match the object casting it. According to what we normally expect from biology, this should never happen. We are taught that an organism begins with a single set of instructions that guide its development from start to finish. Those instructions are supposed to be consistent throughout the body, deciding things like sex very early on, and then applying that decision everywhere. Once that path is chosen, every cell is expected to follow along without question. A body that is half one thing and half another seems to violate that basic promise of order. It suggests that cells can disagree, that development can split into separate directions and still somehow remain stable. When scientists first encountered these organisms, they were not looking for them. They appeared by accident, noticed by collectors, breeders, and observers who thought they were seeing a rare deformity or an illusion. Early explanations tried to dismiss them as damaged or incomplete, something that went wrong during growth. But the more examples appeared, the harder it became to ignore the pattern. These organisms were alive, functional, and in some cases capable of normal behavior and survival. The confusion was not about whether they existed, but how they could exist at all. The idea that a single body could follow two developmental paths at once felt deeply uncomfortable, even to trained scientists, because it hinted that life was less controlled than previously believed. The existence of Gynandromorphs quietly changes how we think about biology. They show that life is not always a single straight line from beginning to end. Instead, it can branch, overlap, and still hold together. They remind us that the rules we learn are often descriptions, not guarantees. Nature does not break when something unexpected appears. It simply continues, unconcerned with our need for neat categories. In the grand scheme, Gynandromorphs suggest that identity in living things is more flexible, more fragile, and more mysterious than we assume. They exist not to shock us, but to whisper a reminder that life is capable of far more complexity than our explanations can comfortably hold. Piezophiles. There is something quietly unsettling about life that thrives where everything else collapses. Imagine a place where darkness is complete, where the cold is constant, and where the weight of the world presses down so hard that steel would bend and glass would shatter. In these places, nothing is supposed to move, grow, or breathe. Yet somehow, life does not merely survive there. It functions calmly, patiently, as if the crushing force surrounding it were not a threat at all, but a requirement. This kind of life feels almost defiant, as though it slipped through a crack in the rules we thought governed all living things. Piezophiles are living organisms that survive and grow best under extreme pressure, usually in the deepest parts of the ocean, where the weight of the water above them is immense. What makes this system feel so strange is how backward it seems. Pressure, in our everyday experience, destroys structure. Too much pressure cracks bones, collapses lungs, and crushes machines. We associate pressure with damage and failure, Yet Piezophiles do not just tolerate pressure, they depend on it. When brought to the surface, where pressure is gentle and familiar to us, many of these organisms slow down, fall apart, or die. It is unsettling to realize that what feels normal and safe to us is lethal to them, and what feels instantly deadly to us is their natural home. According to the basic rules of biology, this should not work at all. Living cells rely on delicate structures. Proteins must fold in precise ways. Membranes must stay flexible but intact. Chemical reactions must occur at the right pace. Under extreme pressure, scientists expected these systems to freeze, deform, or simply fail. Pressure was thought to squeeze molecules too tightly, breaking the balance life needs to function. By these rules, deep ocean environments should have been biological deserts, silent and empty. The first hints that something was wrong with this assumption came slowly and with disbelief. When scientists began exploring the deep ocean, they expected to find little more than mud and decay. Instead, they found thriving microbial communities. At first, many assumed these organisms were fragile visitors that would not truly belong there. But experiments showed the opposite. When these microbes were studied under surface conditions, they struggled. Only when scientists recreated the crushing pressure of the deep did the organisms behave normally. The idea that life could be pressure-loving rather than pressure-avoiding was deeply uncomfortable and took time to accept. The existence of Piezophiles quietly reshapes our understanding of life itself. It tells us that biology is not built around our comfort zone. Life does not aim for ease. It adapts to whatever conditions persist long enough, even if those conditions feel utterly hostile to us. This realization stretches beyond the ocean floor. It invites us to wonder where else life might exist, in places we have already dismissed as impossible. More than that, it humbles us. The world is not designed to make sense to human intuition. It is far stranger, more flexible, and more mysterious than it appears, quietly carrying forms of life that thrive under pressures we can barely imagine, waiting patiently in the dark. Radiotrophic fungi. There is something quietly unsettling about life that thrives where life should be erased. Radiotrophic fungi are not loud or dramatic organisms. They do not move quickly or fight for attention. They grow slowly, patiently, in places that feel wrong. Places filled with invisible danger. Places where most living things weaken, decay, or die. These fungi seem comfortable there, almost as if they belong. Their existence challenges the comforting idea that nature has clear limits, that life knows when to stop, and that certain environments are simply off-limits. Radiotrophic fungi are organisms that can grow by using radiation as an energy source, turning something destructive into something that sustains them. What makes this system feel so strange is how casually it ignores our instincts about danger. Radiation is something we associate with sickness, mutation, and death. It damages cells. It breaks genetic material. It is the reason people evacuate cities and seal off buildings for decades. Yet these fungi do not just survive in radioactive environments. They often grow better there. Instead of shrinking away from radiation, they lean into it. They darken in color and spread more confidently, as if the very thing meant to destroy them is somehow feeding them. At first glance, this feels almost like a mistake in nature. Radiation is supposed to be a stressor, not a resource. Living things usually need light, chemical energy, or organic matter to grow. Radiation does not fit into that picture. It feels more like a force of damage than a source of nourishment. The idea that something could take in radiation and use it productively sounds closer to science fiction than biology. According to what we usually expect from biology, this system should not exist at all. Life depends on delicate chemistry. Cells rely on stable structures, carefully folded proteins, and instructions written in fragile genetic code. Radiation disrupts all of that. It tears through molecules and scrambles information. Under normal rules, prolonged exposure should slowly dismantle a living system until nothing functional remains. Scientists long believed that radiation tolerance had strict limits, and that crossing those limits meant death, not adaptation. The confusion began when researchers noticed dark-colored fungi growing inside highly radioactive environments, including damaged nuclear facilities. Instead of finding sterile ruins, they found patches of life clinging to walls and surfaces soaked in radiation. When scientists studied these organisms more closely, they realized something deeply uncomfortable. The fungi were not just enduring the radiation, they appeared to be using it. Experiments suggested that a pigment called melanin, similar to what gives human skin its color, might help convert radiation into usable energy, somewhat like a twisted mirror of how plants use sunlight. The reaction among scientists was cautious disbelief. Many questioned whether this was truly happening or whether something else was being misunderstood. But over time, the evidence refused to go away. The implications of this system are quietly profound. If life can adapt to feed on radiation, then the boundaries we draw around habitability begin to blur. Environments once labeled dead zones may only be unfamiliar zones. It suggests that life is not just resilient, but opportunistic in ways we have barely begun to understand. It raises questions about life beyond Earth, about survival after catastrophe, and about how flexible biology can truly be when pushed into corners. Radiotrophic fungi remind us that nature does not always share our sense of logic or fairness. What we call destructive may simply be unused. What we fear may be someone else's fuel. And perhaps the most unsettling thought is this: life does not need the world to be gentle. Sometimes it only needs the world to exist. Siphonophores. There is something deeply unsettling about a living thing that refuses to be just one thing. Something that stretches across the water like a single creature, moves with purpose, hunts, eats, and survives, yet quietly breaks the rules of what we think an individual life should be. Siphonophores are dead. Feels unnatural. Siphonophores. There is something deeply unsettling about a living thing that refuses to be just one thing. Something that stretches across the water like a single creature, moves with purpose, hunts, eats, and survives, yet quietly breaks the rules of what we think an individual life should be. Siphonophores are delicate and beautiful, almost fragile in appearance, yet they embody a kind of biological confidence that feels unnatural. They glow softly in the deep ocean, drift like ribbons of glass, and behave as if they are one mind with one intention. At first glance, they seem graceful and simple, but the more you look, the more they feel wrong in a way that is hard to explain. In the simplest possible terms, a siphonophore is a long, floating, marine organism that looks like a single animal, but is actually made of many smaller living parts working together as one. What makes this system so strange is that every visible part of a siphonophore is alive on its own, yet none of those parts can survive independently. Some parts are responsible for movement, gently pulsing to push the entire body through the water. Others are responsible for feeding, armed with stinging cells that capture prey. Some handle digestion, some handle reproduction, and some act almost like support structures, keeping everything in place. Each part looks like an organ, but behaves like a tiny individual animal. It is as if a body decided not to grow organs, but instead recruited dozens or even hundreds of smaller creatures and convinced them to pretend they were one being. This feels unsettling because it blurs a line we rely on to understand life. We are used to thinking that an organism is either one individual or a group. A fish is one animal. A school of fish is many animals. A human body has organs, but those organs are not independent living beings. Siphonophores exist in a confusing middle ground. They are not colonies in the usual sense, because the parts cannot separate and live on their own. They are not single animals either, because each unit begins life as its own entity before becoming locked into the whole. Nature, which usually loves clear categories, seems to hesitate here. According to normal biological rules, Siphonophores should not exist at all. Life is expected to organize itself in predictable layers. Single cells become tissues, tissues become organs, organs become organisms. Groups of organisms may cooperate, but they usually remain capable of independence. Siphonophores break this pattern. Their building blocks are more complex than cells, but less independent than animals. From an evolutionary standpoint, this creates a problem. Natural selection typically favors individuals that can survive and reproduce on their own. In Siphonophores, no single unit can do that. Survival only happens if every piece remains perfectly coordinated. If even one crucial part fails, the entire system suffers. For a long time, this made Siphonophores feel almost like a mistake. How could evolution produce something so interdependent, so fragile, and so difficult to maintain? It seems inefficient. It seems risky. It seems like something that should collapse under its own complexity. And yet, Siphonophores have persisted for millions of years, quietly thriving in the open ocean. When scientists first began studying them more closely, confusion followed. Early observers struggled to even describe what they were seeing. Some believed Siphonophores were single animals with strange appendages. Others argued they were loose colonies behaving in unison. There was resistance to the idea that something could be both at once. The concept challenged how biologists defined individuality itself. Even today, explaining Siphonophores to students often leads to discomfort, because the explanation feels unsatisfying. It sounds like a contradiction, yet it is accurate. As research continued, scientists realized that Siphonophores did not accidentally break the rules. They rewrote them. Evolution had found a way to create a body, not by central control, but by distributed cooperation. There is no single brain commanding the whole. Instead, local signals guide each part, creating a unified behavior without a single center of authority. Movement, feeding, and response to danger emerge from cooperation rather than command. The existence of Siphonophores quietly changes how we think about life. It suggests that individuality is not a fixed property, but a spectrum. It hints that what we call a self might simply be a stable agreement between parts. This idea extends beyond biology. It touches ecosystems, societies, and even our own bodies, which are themselves home to trillions of microbes without which we could not survive. In the grand scheme of things, Siphonophores remind us that nature does not care about our categories. It builds what works, even if it makes us uncomfortable. Life is not always neat, efficient, or easy to define. Sometimes it is a long, glowing chain drifting in the dark, alive only because every part agrees to be something less than whole on its own. And maybe that is the quiet lesson they offer. That survival does not always come from independence. Sometimes it comes from surrendering it entirely. Planarian Regeneration. There are living things on this planet that do not behave the way we expect life to behave. They do not rush. They do not panic when damaged. They do not seem to fear loss in the way most organisms do. Instead, they respond to destruction with patience, order, and an almost unsettling calm. Planarians belong to this quiet category of life. They are small flatworms that live in ponds and streams, unremarkable at first glance, soft-bodied and simple in appearance. Yet hidden inside them is a capability so extreme that it forces us to question whether our assumptions about bodies, identity, and survival are even correct. In simple terms, planarian regeneration is the ability of a tiny flatworm to regrow its entire body from a small fragment of itself. What makes this system feel so wrong is not just that the worm can heal a wound. Many animals can do that. What feels unsettling is that you can cut a planarian into pieces, and those pieces do not die or remain incomplete. Each fragment calmly reorganizes itself and becomes a complete worm again, with a head, a brain, eyes, organs, and behavior. Cut it in half, and you get 2 worms. Cut it into many pieces, and you get many worms. Even a fragment smaller than a grain of rice can rebuild the whole structure. It is as if the animal does not care which part of itself is missing, because every part somehow knows the full plan. At first glance, this feels like something nature should not have allowed to exist. We are used to the idea that bodies have a fixed direction. A head belongs at one end, a tail at the other. We assume instructions are stored in specific places, and that losing those places means losing the information forever. Planarians ignore all of that. A piece taken from the middle can decide where its head should be. A piece taken from the tail can rebuild a brain. There is no visible panic, no confusion. The system simply runs, quietly correcting itself. According to normal biological rules, this should not be possible at all. Most animals rely on specialized cells that know only one job. Brain cells do brain things. Muscle cells do muscle things. Once development is finished, those roles are locked in. If you remove critical structures, the instructions are gone. Identity collapses. Under these rules, losing a head means death. Losing half a body means failure. But planarians do not follow this script. They use a population of cells that remain flexible, capable of becoming anything, even in adulthood. More strangely, these cells seem to share a body-wide understanding of shape and orientation, as if the organism remembers what it is supposed to be. Scientists realized something was deeply wrong with their assumptions when repeated experiments refused to fail. Researchers expected partial regrowth at best. Instead, they watched full animals emerge again and again. Some even altered conditions and saw planarians grow extra heads or regenerate in reversed orientations. The reaction was not excitement at first, but discomfort. This was not just an exception. It was a contradiction. A living system that behaved more like a self-correcting blueprint than a fragile machine. The existence of Planarian regeneration quietly changes how we think about life. It suggests that biology is not only chemistry and parts, but also information and pattern. It hints that identity is not stored in one place, but distributed, resilient, and surprisingly forgiving. If a simple flatworm can remember itself after being taken apart, then perhaps life is less rigid than we imagine. Perhaps our own bodies carry deeper instructions than we currently understand. And perhaps nature has been hiding systems like this all along, waiting patiently for us to notice that the rules were never as strict as we thought.